Hi to everyone, I myself Dr. John Wyson from Amet University, Department of Food Processing and Technology. Today I am going to deliver a lecture on course code UDFP 602, course name Genetically Modified Organism and Genetic Engineering. Today I am going to speak about polymerase chain reaction. Polymerase chain reaction is an exponentially amplification of specific DNA sequences by in vitro DNA synthesis. Polymerase chain reaction. Polymerase chain reaction is nothing but a in vitro DNA synthesis. It is one of the most iconic and popular techniques in a molecular biology field. In recent years, it has been developed by many researchers and it has been carried out by many foreign abroad students. It has been coined by Carey Mullis. In 1983. Generally, for this PCR reaction, it requires three methods, three types. The PCR requires the following to carry out. First one is DNA template. Second one is primer. Third one is enzymes. Whereas here DNA template deals with a molecule to be amplified. So for this PCR reaction, we have to choose the DNA template, which molecule has to be amplified. Next one is primer. The selection of primer is most important here. For entire PCR reaction, the primer is a heart. It's a heart-like thing. So we have to choose the primer according to your study. So here, the primer's main function is annealing. Here in this DNA strands, two strands are there, right? 3' and 5'. If we keep the primer here, it will extend. So next one is enzymes. We need enzymes in the PCR reaction mixtures. Enzymes means what enzymes? Thermus aquaticus. This Thermus aquaticus enzyme has been isolated from the bacterium. So this one is heat stable. So generally the bacteria won't be heat stable, but this bacteria is especially designed for this one, as it will heat stable for 95 degrees Celsius for 1 minute. So these are the requirements for the PCR, polymerase chain reactions. So now we will see the working method. working mechanism. Working mechanism of PCR. So, here also there are many steps in this PCR. First one is denaturation. So, usually denaturation is nothing but melting of DNA, melting of DNA. Here we will use this DNA template here. Though here the reaction starts, the PCR reaction will start initiated in this denaturation process at 95 degrees Celsius for 15 seconds. Hence the strands will be separated. The DNA strands will be here, then the DNA strands will be separated using denaturation method. Next one is annealing. Here the 3' end strands will be extended, strand will be extended. This 3' strand will be extended. Then final one is primer extension. This process will be taken place at 60 degrees Celsius. Sorry, I forgot to tell. Here the process will be taken at 60 degrees Celsius for 45 seconds. Next one is primer extension. Here we will use this adenine, guanine, cytosine and thymine, where DTP will be involved. DTP, DTP, DTP. So DATP, DGTP, DCTP, DTTP. These enzymes will be used, and as I said before, what enzyme? Taq polymerase enzyme will be used. Taq polymerase enzyme is nothing but thermus aquaticus enzyme, and another enzyme is called Wnt polymerase. So both the enzymes will be used. Both the enzymes are the heat stable enzymes, where the bacterium will hold the life for 2 hours here. So this is the working mechanism of PCR. Now we will see the flow chart. First DNA template, right? Here 3', 5, 3, 5. As I said, this one is the DNA template. Next one, annealing. Here the primer will get joined here, right? Three dash, 5, 3, 5. Here also 3, 5, 3, 5. Now primers will be added over here. Next step is primer extension, where here the Taq polymerase enzymes and DATP, DGTP and DCTP enzymes will be added into the PCR tube mixture. Like DATP, DCTP, DGTP, DDTP plus Taq polymerase. What will be the resultant here? Next one is this primer and this Taq polymerase. Primer. Here also primer. Here Taq polymerase. First one, DNA template. The strands will be separated into two primers will be added here. Then after into the reaction mixture, this DATP, CTP, GTP and DTP polymerase enzyme will be added. After that, this two strands will be separated into 4. Four strands are there. 1, 2, 3, 4. Like 3, 5, 5, 3, 3, 5, 5, 3, 3, 5, 3, 5, 3, 5, 3, 5. So in initial DNA template will be in this all involves in one cycle. The whole thing involves in one cycle. Like this. The PCR mechanism will be involved in 35 cycles. So at the end of the cycle, 35th cycle, large quantities of DNA will be isolated. Large quantities, like 1 billion DNA will be isolated from this one. In PCR based techniques, there are many in PCR based techniques. First one is RT-PCR. Second one is hot start PCR. Third one is RFLP. Fourth one is RAPD. Today we will see about RAPD. Before that I will explain about RT-PCR and hot-start PCR and RFLP. RT-PCR is nothing but reverse transcriptase PCR, which involve using reverse transcriptase enzymes. This one is nothing but it will be amplified, hot-start PCR. Here some DNA will be restricted. Now we will see about RAPD, random amplified polymorphic DNA. RAPD is nothing but short oligonucleotide or arbitrarily primer. in the DNA fragments, in this DNA fragments 3 dash, 5 dash and 5 and 3, when this RAPD process involved, randomly this arbitrarily DNA will be cut and isolated. Like what we see in the PCR, the mechanism involved like isolation of PCR, isolation of DNA, primer, annealing, extension, enzymes. After that, the last one is very important. After isolation of DNA, in RAPD mechanism, how we can detect this PCR by the visual method. We can see the PCR isolated DNA by gel electrophoresis. Using this gel electrophoresis method, we can view the DNA isolation. Now we will see about the gel electrophoresis method. What do you mean by electrophoresis? Electrophoresis is nothing but migration of charged molecules by applying an electric field. Migration of charged molecules by applying an electric field. Here we have the formula like V equal to E by F, where E denotes the velocity of the current, E electric field applied, F fractionation, fraction of the molecule, fraction of the charged molecules. Now we will see the working mechanism of electrophoresis. Working mechanism of electrophoresis. For the gel electrophoresis runs, we have to prepare certain chemicals for that. First, we have to prefer the pH. pH should be 8.8, then pH 6.8. Two gels has to be prepared. First one is stacking gel. Next one is separating gel. Here we have to use distilled water 1.9 ml. Next one is tris buffer 1.7 ml. Next one is acrylamide 500 ml. APS is nothing but ammonium persulfate. We have to take 500 ml. Then last one is temad 50 ml. First we have to prepare the stacking gel, next separating gel. Here we have to use distilled water as 1.5 ml, tris buffer, tris pH 6.8, 1.3 ml, acrylamide 500 ml, APS ammonium persulfate 250 ml, then temad 25 ml. First we have to collect this, first we have to prepare these things according to the molecular formula and a ratio. Like tris buffer 1.7 ml means we have to take like 9.1012 gram of tris buffer and 500 ml of distilled water. Next one is acrylamide. Acrylamide is the same like that, 7 gram of acrylamide plus 3.2 gram of bisacrylamide. Then we have to make it up to 50 ml. 50 ml of distilled water. Next one is ammonium persulfate. Ammonium persulfate is like 0.05 gram of APS. We have to dissolve in 5 ml of distilled water. We have to take the gel board, like it will be like that, this one. First we have to pour the stacking gel. Next one is stacking gel will be like this here. Next one, separating gel. After pouring this one, after pouring the separating gel, we have to keep the comb like structure. Comb, the gel comb, we have to keep here and wait it for 30 minutes like this. After this, we have to pour the sample here. Sample ratio is like we have to take, we can take any sample like which are rich in protein, like 100 mg of sample plus 1 ml of water. We have to mix it, centrifuge, then we have to collect the upper layer. After pouring the sample inside this, you have to pass the electric field. Nothing but current. First we have to keep in 100 volts. Gradually 200, 300, 400. Depending upon the current, the sample, the pore size, the protein molecule will migrate here like this. See. like this. At last, you can see a thick band here, thick lines here. After this process overs, we have to put it in a staining solution. We have to prepare the staining solution using glycerin plus methanol plus ethanol. You have to prepare this staining solution and keep it for 1 hour in a dark room. Once this gelling property is over, we have to place this gelling properties inside this staining solution, like here. After 1 to 2 days, you can see the bands here, the thick bands. According to the DNA template size, in the first stage we have been included the DNA template, right? Like 100 to 5000 base pairs. Depending upon that 100 to 500 base pairs, it will be it will be isolated, like 100 base pairs here, then 1000 base pairs here. So this is the overall mechanism of PCR and gel electrophoresis method. First, initially we have been isolated PCR using a DNA template, primers and annealing. First we have kept the DNA template, then it has been extended by primers, then last one we are adding that Thermus aquaticus bacterium enzymes and kept it in a PCR cycles for 35 cycles. So after completion of 35 cycles, we can get the large quantities of DNA here. So using this DNA, we can isolate this electrophoresis method. Whereas in this electrophoresis method, we can use this separating gel and stacking gel as a key tool inside this, and we can isolate this DNA. So this is the overall mechanism of PCR. Thank you.